Recording. Share my screen. This is different. Change that. All right, please note my website where you can find the laboratory material as well as everything else. We will be discussing lab module 00 on lab safety today. The worksheet lab 00 is due Friday, September 24th at 8 a.m. I'm going to take attendance on this worksheet. So make sure you turn in this worksheet by 8 a.m. On Friday, or else you will be dropped from the class. Any questions about any of that? If there's no questions, let's go on to lab 00. Oh, I got to talk a little bit about the syllabus. Sorry, I forgot about that. Oops. Let's go on with the syllabus. Let's shut it down. No, there it is. No, that's not it. So we'll be meeting from 6:30 to 8:20. After 8:20, I'll definitely log off. What we'll do is I'll talk about the lab, and then I will let you guys work on the lab. And if you have questions, I will be here to answer those questions. That's the way I work the lab. And then at 8:20, I'll log off. Now, I will log off earlier if all the students log off before 8:20. So if you have a question, make sure you get in earlier than 8:20. There's one part I want to talk about in the syllabus. Obviously, it's a part of your grade, a large part. Laboratory safety rules. You should read over this material. I should describe part of it. Safety is one of the most main skills that the nursing programs want students to learn, and then other students need to learn it as well. Safety is of primary importance in the lab. The attitudes and actions determine the level of safety. Never take shortcuts which risk safety and accuracy. So follow these rules and you will do well in the lab. Normally, you cannot take things into the lab and take them to your lab bench. They must remain in your bag and in the cubby holes, which, although it's inside the room of the lab, we consider the door and then the cubby holes outside the lab. And then once you walk into the lab, we consider that the lab. Does that make sense? So any personal items, if they come to your lab bench, you cannot take them out. They must remain in the lab until they get disinfected. And to be disinfected, it has to either go in the autoclave or put in something like a bleach solution. And if you take your phone and put it on the table, the instructor would grab it and it will be disinfected, and then it will be given back to you and it will no longer work. So all electronics should never be used or even used in the lab or taken into the lab proper. They can be left in the cubby hole. So all personal items, are in the cubby hole or left out of the lab. That includes your coat, your bags, anything that is brought to the table in the lab or in the lab proper has to stay in the lab. Paper and writing instruments are provided for you, but you could bring them into the lab as long as you realize they have to stay in the lab until they are disinfected. You cannot take anything home until it's disinfected. Cell phone use is prohibited. If you wanted to, I don't know, answer a call, what you need to do is stop what you're doing, go take off your gloves, wash your hands, answer the phone, in which case you'd have to call them back because it'll be too late. Go outside the lab, talk on the phone, come back in the lab, put the phone away, wash your hands, put your gloves on before you can go back to working in the lab or just in the lab. Any questions about any of that? All right. We won't be in the lab, so it shouldn't be a problem, but this is the procedure. What we're doing with these rules in the lab is trying to prevent bacteria that are in the lab from leaving the lab and stuff that is in the lab being taken home to not get contaminated and being taken home. This is a concern, especially at Clark, because of course this is over 10 years ago now. Somebody took home salmonella from a microbiology lab and then made a meal for their significant other and gave the significant other salmonella. And they got sick and then went to the doctor, and the doctor did a test on them and found it was a strain of salmonella that came from Clark College. And so we set up these guidelines to prevent bacteria from leaving the lab and making anyone sick. Any questions about any of that? All cultures are considered to be potentially pathogenic, so you should keep them, all your material, away from these cultures. Obviously, no eating, drinking, smoking, gum chewing, or other such oral habits are allowed in the lab, and you must wear gloves while working in the lab or in the lab at all times. We're talking about nylon gloves. You must wear a lab coat when you're in the lab, as well as gloves. And if you're working with a splash hazard, that would be bacteria in a liquid culture or some liquid like a dye where you can splash it into your eye, you must wear goggles. But only while you're working with a splash hazard do you have to wear goggles. But the lab coat and the gloves have to be worn all the time in the lab. You must also have closed-toed shoes. I'll show you my slipper. This would be allowed. Can you guys see that? This would be allowed in the lab because it's closed-toed. There are some fancy women's shoes which have an opening at the toe. Those are not allowed. Students always assume that we want closed-toed shoes to protect your feet from drops or breaking glass or dropping a Sharpie, not a Sharpie, a scalpel or something like that. That's not true. We want your closed-toed shoes to offer protection in case you spill your culture of bacteria. And closed-toed shoes give some protection, not necessarily a lot, but it does give some protection. Any question about any of that? Cleanliness is a virtue in the lab, and you need to clean down with disinfectant the lab surface you're going to work on before you work on it. Normally you do that at the start of the lab, and then before you go, you need to clean down that surface again. And of course, before you enter the lab, you have to wash your hands, put on gloves, put on the lab coat, and then before you go, you take off the gloves, take off the lab coat, wash your hands. Accidents should be reported to the instructor. I'll let you read about what you do because it won't be in the lab. We use a Bacto incinerator. You'll actually see a picture of it in the future lab for sterilizing your laboratory instruments. You could also use a Bunsen burner, but we can put it directly into the fire, but we have a Bacto incinerator. Cultures never leave the lab. If you have a cut, it should be Band-Aid. On your hand should be Band-Aid, and then you put the glove on. You do need to wash your hand even if you have a cut, so I guess you should wash your hands, put on the Band-Aid, put on the glove. Everything has a proper place for where it is discarded. Where is that in here? Let me just state that we have a container, it's usually a beaker or something like that, for putting glass slides. They go in that, and it usually has a disinfectant like bleach in it. There's also a little beaker or container, plastic container, for putting used Q-tips. They have bacteria in them, and they've got bleach in them. Cover slips we do not wash and reuse. So we have a separate container for cover slips. And usually you just put those in the little beaker with bleach, and then those will be just thrown away. But the slides and all the other material will be, at least the glass material, will be put in disinfectant or autoclave, and then washed, and then reused in the lab. Any questions about any of that? Other material that you throw away, if it is plastic or paper and contaminated, you put it in an autoclave bag or a biohazard bag. And I use both terms because the biohazard bag will be sealed, not sealed, but shut, and then put in the autoclave, sterilized before it's discarded. Glass, you put it in a special container or bin, like glass test tubes, remove the label from them, and then just leave it there. And that will be autoclaved and then washed and then reused. Glass never goes in the autoclave bag. And that includes slides, Petri dishes, which we don't use glass ones, and pipettes. What else do we use as glass? Beakers, things like that. They have a special container that you put it in so that it can be autoclaved and then washed and then reused. So only plastic, Petri dishes, paper that's contaminated with bacteria. What else? Plastic pipette can go in the autoclave bags. Any question about that? Glass, if it's contaminated, it should go in a special container so that it can be treated and disinfected before it's washed and then reused. Material like paper or an agar plate that you're not using, doesn't have contamination with bacteria, those, if they're not contaminated, can go directly in the trash can. Any question about any of that? Broken glass, we have a special container for putting broken glass in. And if it is contaminated, it should be disinfected with disinfectant first and then put in the broken glass container. So all supplies needed by the lab are supplied in the lab. All right, any question about how we use the lab and the rules of the lab? So today we'll be talking about Lab 00, laboratory safety. Yep, that's the wrong one. There we go. You're not going to be working in the lab, but you do need to know the material that is presented in the lab modules. So upon successful completion of the lab, this lab student should be able to distinguish between safe and unsafe laboratory practices, understand where relevant safety equipment is located in the lab, and that one, you're not actually going to be in the lab, so it's not a big one. Understand how to safely dispose of contaminated materials. Mainly if they're contaminated with bacteria and they're not glass, you put it in the autoclave bag, also called the biohazard bag. If it is glass, you need to put it in a special container. And we have different special containers for different glass items. Like there's one container for putting slides, there's another container for putting test tubes, and there's a third container for putting pipettes. You should wear proper attire when you're in the lab. You're not actually going to be performing any of these labs, but we're going to go over them, and you need to do the lab and understand the lab. And today's lab will be teaching you about creating a safe laboratory environment. You need to wear a proper lab coat, and those are actually supplied by the lab, although a few students do buy it from the bookstore, in which case we autoclave it at the end of the term and then the student can take it home. But everyone else, those labs coats are left in the lab and they're just reused term to term. We do supply disposable gloves. You need to wear closed-toed shoes. You need to wear goggles when there's a splash hazard. And let me think. While you're on campus this term, you need to wear a mask. Of course, we won't be on campus, but that's what's happening on campus. And then all of those items will be removed before leaving the laboratory. I wonder what they would do with the mask. My guess is you take off your mask that you brought into the lab, and then you put on a new one in the lab. And that one in the lab will probably stay in your lab drawer, or else we would resupply a new lab mask for every lab. I don't know how to do that. We'll have to worry about that whenever I teach the lab in the lab on campus. Although winter term, I'm the only instructor who's going to be teaching it online. All right, if you have long hair, it should be tied back so there's less fire hazard. Absolutely no food or drink being eaten in the lab or use of electronics. No personal items are allowed in the main part of the lab, including cell phones. This does mean you can take whatever you're wearing. If you're wearing a watch, you can take it into the lab, cover it with the glove. If you're wearing jewelry, you don't have to remove that. You can take that in the lab. Just don't touch it while you're in the lab. And then all personal items are stored in the cubby hole where you put your book bag. It's near the lab door. So proper laboratory attire. As I mentioned, you need to disinfect the area before you start and then before you leave the lab. This is actually a picture of our lab, by the way. What do you think about that? Is that our chairs? I don't think we have wooden chairs, but I don't remember. I should, but I don't. There is a biohazard container, so everything that's contaminated with bacteria should be put in there, except glass. If the glass is contaminated, it goes in a special container for putting glass. And also, I guess, swabs, we have a special container for swab. Although if you put a swab in the biohazard container, that's not a problem. But we have a container for putting slides and cover slips, as well as beakers and a container for test tubes. Generally, we don't use beakers in microbiology or containers like this, but you do use glass test tubes. Report all spills and accidents immediately. Cover the spill with disinfectant, and then put paper over the spill so the disinfectant gets spread or wicked by the paper. Then allow it to soak for 15 minutes and then discard in the biohazard trash container, unless it's got broken glass. The broken glass will go in the broken glass container after it's been disinfected by the disinfectant you added and let it soak for 15 minutes. No mouth pipetting, no chewing your nails or anything else like a pencil while you're in the lab. But we don't use a flame like this, but we do sterilize our instruments in a Bacto incinerator, and that's what our Bacto incinerator actually looks like. And yes, that's red hot, the metal inside and the stone inside. You should know your safety equipment that's in the lab. There is a fire alarm, which has never been used in my lab. A first aid kit, where you can find bandages, and those have been used in my lab. The spill control station, which has never been used. I'm not even sure what's in it. Oh, I think that's charcoal, and you put that on the spill to inactivate whatever the spill is. The eye wash station, which has never been used in my lab, and a shower in case you spill something toxic on you. You put yourself under the shower. There's usually a fire blanket, which is not shown. And then back behind the spill center is the fire hose, which you can spray water with. I don't think we have a fire hose in our lab. Oh, we do have a fire extinguisher though. So you should pre-read the labs. That's not terribly important for this lab, but in the future, pre-read the lab, and then you'll be ready to go at the start of the lab. So the first 2 pages of this document detail the safety rules that apply to microbiology lab at Clark. You're expected to know these rules and you'll be tested on them in the quizzes. But the good news is you're not actually going to be in the laboratory, so there's no way to follow these rules. I already mentioned don't take shortcuts, and I've talked about everything should be left at your cubby hole in your book bag. If you bring it into the lab, it has to stay in the lab until it's disinfected. Only microbiology students are allowed in the Clark College lab and thus permitted by the instructor. We talked about cell phones and private supplies. Consider all cultures to be potentially pathogenic, so handle them with gloves and with care. If you were to come into the microbiology lab, you should monitor yourself for possible infection with a bacteria, such as looking for diarrhea, fever, abdominal cramps. That's not so important in this class because we won't be really handling live bacteria. We talked about lab coats. I talked about goggles and cleanliness, fire extinguishers, accidents. I already talked about all of this stuff. Oh, here's something we haven't talked about. All microbial cultures in the microbiology lab have to be labeled completely with your name or your initials, the date, the nature of the specimen. So if it's E. coli, just write E. coli. If it's something you don't know what it is and you got it from exposure to the air, say air exposure. and then the medium that's used. Any question about that? So every culture, every petri dish, every test tube should be labeled with your name, the date, the nature of the specimen, and the medium. We'll talk about media a little later in the term, but write down what the medium is. Optional, you may also put the temperature that you incubate the organism, but I don't require that. It's optional. Old specimen should be discarded appropriately. If it's in an agar plate, put it in the biohazard bag. Glass slides should be put in the glass slide container. Cotton swabs, assuming they're contaminated with bacteria in the swab container. Pipette should be in the pipette container. Glass test tubes in the test tube container. Remove the tape that you had to label the test tube before you put it in the container. Generally, students work in pairs when they're in the lab. Obviously, you won't be doing that because we're going to do it online. I already talked about that too. All right, that's it for the rules. Any questions? That's it for lab module 00. So work on the lab worksheet 00. Let me put it over here. I'm not there anymore. Answer the questions. I didn't quite understand what was going on here. And apparently it's viewed differently than when it prints. If you were to print it out, there would be more information than shown here. And so I just put the questions down here and said, ignore the above. Rule number 1 in the lab, what do you always wear for safety before you enter the lab? And there's 4 asterisks, so I expect 4 answers. If you don't supply 4 answers, you only supply 3, you'll get the 4th one wrong. Rule 2, after you enter the lab, what do you put on to keep yourself safe while in the lab? So this is what you have to wear before you come into the lab. This is what you wear when you enter or after you've entered the lab. Okay, so rule 2 should be different than rule 1. I will tell you that generally in rule 1, you have to have closed-toed shoes, for example, before you enter the lab. You don't put them on in the lab, although I guess you could, especially if you were wearing sandals, like in the summer. You could bring your closed-toed shoes in the lab and put them on in the lab. But normally most students will have them already on. Got that? So what's something you put on while you're in the lab? And you do not put it on before you enter the lab. Anyone answer?
Protective eyewear.
Yeah, that would be one. There's a couple of the things. There's at least 3 things. But
Gloves.
Yeah, gloves would be another one. Remember, with protective eyewear, you only need to wear it when you're working with the splash hazard. And once you're done working with the splash hazard, you can take off the goggles. All right. You need to look up about what a lab safety data sheet is online, and then answer this question. Each chemical in the lab must have safety data sheet that describes, what does it describe? Know the location of the safety equipment, and then list the different safety equipment. An eye wash station is one, okay? So I'm not asking for the location of it. You're not going to be in the lab. You're not going to know the location of it, but you should know that there's an eye wash station, okay? And you need to supply 1, 2, 3, 4, 5 things. And then what can you take into the lab? And can you eat and drink in the lab? And so there's 3 answers. So what can you take into the lab? Well, you can take your watch, for example. If you want to use a specific pen or pencil, and as long as you leave it in the lab, you can take that into the lab. And can anyone answer? Can you eat and drink in the lab?
No.
All right. At least one item of laboratory attire you must wear in the microbiology lab, and then one item that is allowed in the microbiology lab. And then submit this by 8 a.m. Friday of week 1, or else you'll be dropped from the class. I'll use this to take attendance for week 1. All right, that's it for lab 1. I doubt that I will stay here to 8:30 because this won't take you a long time to finish this lab. But when the last student has logged off, I will log off as well. So go ahead and start working on the lab. Any questions?
Is the information for rule number 3 for the SDS going to be in our microbiology book, or do we have to go online and look that up?
I think you'll have to go online to get that information. The SDS information may be in the complete textbook, but in the textbook that is made for Clark College, I don't think it's in there.
Okay, thank you.
Okay.
Question. So we just ignore you said just ignore the picture, right? We don't do anything with it.
Yeah, if you print it out, you'll get the same questions that I've got below.
Okay. Okay. Perfect.
All right, hopefully I recorded this. I'm going to stop recording. Stop sharing.